I am the Research and Conservation Programs Coordinator at African Lion Safari. I am also an adjunct professor at the biology department at Queen's University. The work that I do largely focuses on how ex situ populations of animals can assist with conservation of species at risk. And while African Lion Safari is best known for its non-North American species like elephants and cheetahs, my work focuses a lot on how our zoos in Canada can actually assist with the conservation of Canadian at-risk species.
Great. So can you tell us a little bit about the Massasauga rattlesnake and why do you think this is an important species for Canada?
Wow. So yes, the Massasauga rattlesnake is only found in Ontario. It is the only venomous snake that we have. There used to be another rattlesnake called the timber rattlesnake, which has since been extirpated. Massasauga actually isn't a highly venomous species. So if you got bit by it, you definitely do want to get anti-venom, but you will not die within minutes, as you would see on, say, Indiana Jones or something like that. The species has been here for a long time, longer than us. Unfortunately, it likes many of the areas that we as humans like in Ontario as well. And so if you look at its historic distribution versus its current distribution, it has had a shocking reduction in range size and in population size. It occurs in two different types of habitat in Ontario, which makes it interesting from an ecological standpoint. One of the types of habitats is prairie, which is the most threatened ecosystem in North America. We have a very small remnant population of prairie in the Windsor region, the Ojibwe prairie. And so Massasaugas are part of that ecosystem. And so in a way, you could consider them, unfortunately, a representative of how little we have left of that particular type of ecosystem. The snake itself, though, it's a very fascinating species in that it does seem to be adaptive to different conditions. It has a really neat symbiotic, the perfectly good term, but relationship with a crayfish. So it actually uses crayfish burrows as overwintering sites. And there are actually some interesting stories going back to First Nations cultures and how the rattlesnake is essentially it's a reminder not to overuse. So it's a really, really neat species. If you've ever seen it in person, it actually has a very tiny little rattle. Unlike when you think of a rattlesnake, you think of the Westerns with this great big rattle, and you look at the little rattlesnake, the little rattle on a Massasauga, and you think, really? Does that do anything? And then it actually does. It's just a super neat species that is incredibly misunderstood.
Amazing. That's an amazing description. Thank you for sharing that. So going into now the genomics side of things. So can you share about the work with the Massasauga rattlesnake and the Canada Biogenome Project that you're leading?
So the Canada Biogenome Project has a focus of creating high quality annotated genomes for a suite of about 400 species. Included in those species are those are at-risk species in Canada that have a recovery strategy that has mention of ex situ management possibly being useful to assist. So an example of that, a species I work extensively is loggerhead shrike. It's a small, critically endangered bird found only in Ontario. And the population here at African Lion Safari and at the other breeding facilities is used to augment the wild population. And then analyses have shown that because of this, we have bought time and the species has not been extirpated in the wild, which is really important. But really interestingly, we have also found out through genetics some new things. And so, for example, again, loggerhead shrike ends up that it was a previously undiagnosed subspecies. So we have a very special component of the genetic diversity in this species here in Ontario. So the having that high quality annotated reference genome really provides a whole bunch of useful management tools and better understanding of the ecology of the species that we're working at. And therefore it can improve, in my view, the recovery efforts for the species. So having the reference genomes is one thing. That's the focus of Canada Biogenome Project. My own work is to go a step further and actually create genetic data that can be used to help in the management of populations. So things such as inbreeding are very important when you have an ex situ population. And the normal mating, so matings are put together based on a stud book, which is basically based on pedigrees, but pedigrees are not necessarily the best way to put these pairings together. So, for example, you could look at runs of homozygosity instead, and they're doing some super cool work with other species where you can say these two individuals have large areas that are homozygous in their genome, but if we match them up, we're not going to overlap them. So it will kind of make sure that we're not losing diversity with genomic tools. So basically, yeah, my work is to sort of do downstream from having the genome. It's improved by having a genome, but it's to develop genomic tools that can assist us in population management of ex situ, but recognizing that ex situ and in situ under the IUCN's one plan approach are combined.
Very interesting. Thank you. So in terms of the plans then for the Massasauga rattlesnake, can you share a bit about that? Why was it chosen for the Canada Biogenome Project specifically?
So there's a couple of reasons behind it. So we had, I'm a co-founder of the Canadian Species Initiative, which has a mission of trying to adopt the one plan approach in Canada more so than it has been done. So doing it in a systematic way, following the IUCN Species Survival Commission Conservation Planning Specialist Groups processes and workshops. So there's, for example, ex situ conservation assessments can be done that clearly look at the different roles that an ex situ population can play. And there's 11 different roles that have been identified, and saying, okay, if this is the threats to the species, then can we use an ex situ population to help address those threats? And if so, how do we do it? And then what does that actually look like? So, so for Canadian Species Initiative, we are focusing on those species initially that do have a recovery strategy that says, informally, we think ex situ could assist, but hasn't been through this formal process of saying exactly how they could assist, and may or may not have that population already. Massasauga rattlesnakes have both a recovery strategy saying that ex situ is a very important component of the overarching recovery activities, but it also has an existing population. And because of the difference in where the species occurs, there are some questions that really can only be addressed using genetics, or best addressed using genetics. For example, local adaptation. So are the snakes in the Ojibwe area genetically different in a way that fitness would be impacted if you took snakes from a gray Bruce and you released them down there? So between having a fairly new population and having a population being so closely tied with the in situ recovery efforts, it seemed like a really good focal species to begin with. As well, I have to say there's been really amazing reception from the recovery implementation group to the integration of genomic tools. So just having that welcoming, open audience that had a really well established group of different rights and landholders and interest groups made it just a really good choice for a focal species.
That's great. And there was recently a workshop held. Is there anything you can share about that? I think that was the first time you convened all the different stakeholders. So can you explain maybe what the purpose was for that and what maybe are the next steps that you're just currently discussing for that?
Sure. Yes. So specifically my work with the Canada Biogenome Project, through the funds that have been secured through that project, I've been able to hire a couple of research assistants through Queen's University. The focus of our work over the past and the first year of the project has been on trying to provide the structure and the framework to help the end user of the genomic tools figure out how the tools could be used and what are the most important tools. Genetics can address tons of different questions, but if that question has a conservation relevance or not is a separate question. So, you may be interested in how genomics relates to the underlying coloration in a species. Does that really have conservation value or not? So the idea was that we wanted to provide the end user and layperson terms information that they could understand about genetics and how genetics could be used, and then provide a process whereby we could help them match up basically a genomic tool with a threat and a recovery action. And so we spent a year working on trying to figure out what that framework would be and how to actually do that through a series of short seminars. We had a short game that tried to help people understand data and how data is, genomic data is put together. And then we had facilitated breakout groups where we took the existing recovery strategy for the species, and based on our expertise, such as it is, as geneticists, we had said, we think that genomics can assist with these things that you have said are of interest to you. And then we had discussion about that where they got to vote and say, well, of those recovery actions or target goals, these are the ones that are most important to us. And then that was how we steered the conversation. So it was an intensive one-day workshop. It was fantastic. We're hoping to use this framework for other species, so to kind of keep creating similar workshop processes. And again, Massasauga is our guinea pig, and wonderful people to work with. So thank you for them. So what we're doing now is we're going to summarize all the results from the workshop and the breakout groups and the feedback that we got. And then we're taking another step from our decision-making framework that we presented and went through, and suggesting specific genomic methods. So there is, for example, whole genome, you could do a whole high quality annotated genome, or you could do a reduced representation sequencing of some sort, ddRAD-seq or something else, pool-seq. And so we're going to provide them with an explanation of what these different methodologies are, pros and cons, including costs of that approach, and then suggest to them basically a program for genomics for their species that will address their high priority research questions. And hopefully we'll be doing that later this summer.
Great. So I guess just in terms of the Massasauga rattlesnake, what are you most hopeful for, I guess, in terms of what could be the next potential steps now that you will have the reference genome from this project? And what do you think, just in general, conservationists who might not be familiar with the application, what do you think one of the most important things that they should consider in their work in the use of genetics and genomics for their conservation efforts? So two different questions, Beth.
Two different questions. So for the first part, for Massasauga rattlesnakes, we are actually, we're actively working now to get permits in place to do blood sample collection from all the individuals in the ex situ population. And then we will be doing some sort of genomic assays down the road and begin to provide some actual results of the assays back to the recovery team to help them address the questions that they want to do. So of those questions, some of the biggest and most important that have come from the workshop was figuring out how best to pair different individuals. So, as I said, you often do it on a pedigree basis, but there's some assumptions around that, such that you're saying every founder that comes into the population is equally important. And important by this means that they are equally unrelated to each other. They have equal levels of genetic diversity within their individual. So, essentially everybody is the same. But that's actually in reality not the case. And so if we could start to provide a method whereby it doesn't necessarily have to be yearly, but on a regular basis, we can actually screen the genetics in the population that we have in our human care and make sure that we are meeting our goals for management, whereby they can be sustainable over the longer term. That will be huge, because we'll actually be able to quantify the management goal. In the bigger picture, being able to have a genomic tool will really help with that integration of in situ and ex situ. And when you think about it, if the purpose of your breeding, having an ex situ population is a conservation breeding program to augment the wild population, you're putting animals out there and you might be able to track that animal. So in the case of loggerhead shrike, every young shrike that goes out into the wild has a leg band. Birds are normally identified by leg bands. And so we can track that individual. We can't track their progeny though. So after a first generation, and then more generations down the road, we really don't have a good understanding of the actual impact of our management efforts. Genomics is a tool that would allow us to do that in a way that no other tool can do that. And then hopefully in an adaptive management scenario, we'd begin to sort of be able to understand a little bit and tweak our management. So say, for example, we're finding that a certain age class is more susceptible based on the release of the individual and the mark recapture data we get back from genetics. We could maybe perhaps hold individuals longer to get them over a critical stage of development, like head starting programs do. We can also make sure that the representation of lineages that we're putting back in the wild is correct based on what was historically there. And if needed, bring in additional lineages, breed them in and put them back out again. So it really would revolutionize the entirety of the management and conservation efforts. And I think in the long run, make them much more successful and hopefully cost efficient as well.
That's amazing. Thank you so much for sharing all that. Is there anything else that you would like to share? Any final thoughts?
No, I get so excited about the potential value of genomics and how it can really assist us with conservation. And especially in Canada, where we have a lot of populations that are at the edge of their range. Local adaptation is something that is very important. If you think about climate change and how things might be changing, it's just the data that we can get from genetics is really going to be necessary to help us address the biodiversity crisis that we've got and be better managers of what we've got, and hopefully help us better appreciate and understand what we've got. As I said, with loggerhead shrike, we never knew that we had, a specifically adapted to our area subspecies until we looked at it with genetics, and then we were kind of blown away. So it's really exciting. I really love working with young researchers, and I really hope we can get this tool, into the people that are using it, and make some great progress.
Amazing. That's great. Well, thank you so much for taking the time to share this, and congratulations on the work. It is very exciting. So can't wait to see all that comes out of it, and we'll definitely continue to share it through CGEn. So thank you for taking the time to speak with me today.
Great. And my thanks to CGEn. If you guys weren't doing what you're doing, I couldn't do what I'm doing. So I really, really appreciate your efforts and your interest in it. So thank you.
Thank you. Take care.